Well, I want to thank SLC6A1 Connect for the invitation, especially Amber and Sandy and Laura, Kim, everyone who's done all the great work to get the organization to where it is now. I'm going to talk today about kind of an update and some new data following on from the work that we've been doing on a potential new drug target that's on SLC6A1 that's related to, maybe related to the action of Ravicti. So this is a slide that I've shown in the past, but I think it's worth just looking at to kind of bring everyone up to speed on what are we really looking at in terms of a pathological mechanism in SLC6A1 mutations. And what we've been studying is a loss of function mutation in SLC6A1. In this case, it's S295L. And this is a complete loss of function mutation. And of course, in patients, because it's a de novo mutation, it's a heterozygous loss of function. And when we look at the mouse model of this mutant, what we can see is that if you compare the amount of uptake in the wild type, is the column on the left, the heterozygous mice have less uptake. This is an assay using radioactive GABA to study uptake of GABA into the brain. And then when we look at homozygous mice, there's even less uptake. And what's important is if you look at this last column, that's the wild type brain tissue that's incubated with this SLC6A1 selective inhibitor, SKF. And that blocks all of the SLC6A1 mediated GABA uptake. And so you can see that the amount of uptake in that treated group, in the treated tissue, is the same as the homozygous. And the heterozygous brain tissue has intermediate amounts of GABA uptake. So this makes sense in terms of a gene dosage effect of what we see as loss of GABA uptake. But now importantly, you can notice these are background subtracted, and you can see that even in the complete loss of SLC6A1, there's still quite a bit of uptake remaining. There's only about a third of the uptake is lost in the mutant animals. And I'll talk a little bit about what we think that remaining uptake is. So if we look at this kind of gene dose dependence of loss of GABA uptake, the total GABA transport is reduced in the het and the homozygous mice. Now, what's interesting is if we look at the amount of uptake that's mediated by the other major GABA transporter in the brain, which is SLC6A11, also known as GAT3, we can study that by another selective inhibitor called SNAP 5114. And what you can see is that in the wild type brain tissue, there's very little effect of blocking SLC6A11. But in the heterozygous and homozygous mutant SLC6A1 mice, there is an effect of this GAT3 blocker, which means that SLC6A11 or GAT3 is providing partial compensation for the loss of SLC6A1 in the disease. And this is a topic for another talk. I'm not going to have time to go into it. We think we understand why this is going on, and it's kind of a lucky coincidence that it's sort of a mechanism that the animals have to help offset, compensate for the loss of SLC6A1 in the disease. So what are we really talking about in terms of the direct mechanism of the disorder? If we consider what determines extracellular GABA levels, which is also known as GABA tone, it's determined by a balance of the release of GABA from neurons and astrocytes and the reuptake of GABA into neurons and astrocytes. And the release is mediated by synaptic release of GABA when neurons are communicating with each other. Inhibitory neurons release GABA. And it's also caused by some tonic release of GABA through other channels. I'm not going to talk about them, but in astrocytes especially, there's a molecule called BEST1 That's a channel that allows GABA to come out. And these mechanisms of release of GABA are counteracted by the reuptake of GABA into neurons and astrocytes through the GABA transporters. And as I mentioned, there's SLC6A1, or GAT1, and SLC6A11, GAT3. These are the two major GABA transporters in human brain. So why is the tone of GABA, or the extracellular level of GABA, important? I'm not going to go through all the details of this slide, but the basic idea of this is that there are really complex actions of tonic GABA, of ambient GABA on neurons in the brain. And those are mediated by actions on GABA A receptors that are found on neurons. And these tonic GABA currents mediated by GABA A receptors cause currents both in the presynaptic synapses of excitatory as well as inhibitory neurons. And they're also present on the postsynaptic neurons in dendrites and in postsynaptic densities. And to make a long story short, this slide goes into some kind of biophysics that are beyond the scope of what I want to talk about, but the effects of raising GABA, which occur in SLC6A1, can lead to multiple types of effects, including increases in excitation of excitatory synapses or inhibition of excitatory synapses. And the same is true at inhibitory synapses. They can either be excited or inhibited. And that's because the chloride concentrations in different parts of neurons in different regions of the brain can be very different, which has different effects as a consequence of opening GABA A receptors. Also, the postsynaptic effects of increased GABA tone cause something called shunting to occur, which basically inhibits the signaling that can occur in neurons. It will blunt the synaptic potential amplitudes. It'll change the time course of the synaptic potentials. And these kinds of changes can have all kinds of complex effects on signaling, on synaptic transmission, synaptic plasticity, as I'll show a couple of examples of in a minute. So we can see directly that GABA tone is changed in the mutant animals if we record the GABA A receptor activation in either wild type neurons, and we can do this by adding a GABA A receptor blocker, picotoxin, and we can look at the offset of the standing current in the response to GABA A receptor blockade in the heterozygous and homozygous animals. And the statistics are shown here. What we can see is that there's a greater standing GABA A receptor current in the heterozygous animals and even greater in the homozygous animals. And we can infer from the change in the amplitude of that standing current that the GABA concentration change in the heterozygous mutant animals is roughly twofold. So it's actually a relatively modest increase compared to what we would predict from theoretical calculations of the effective loss of half of GABA transporters, because there's a nonlinear relationship between the steady state levels of GABA and the concentration or the density of transporters. And part of that might be due to the fact that GAT3 is functionally upregulated. So one of the things that happens, I mentioned there's this phenomenon known as shunting that really causes a decrease in the excitation coupling to spiking that occurs in neurons. So we can see this if we compare wild type, heterozygous, and homozygous animals, and these are whole cell patch clamp recordings of neurons in the hippocampus. If we inject equal amounts of current into these three genotypes, you can see that the spiking is more robust in the wild type, and it's less in the het and even less in the homozygous. And if we plot the current injection versus the spike frequency, you can see that there's this shift where more excitation is required in the mutant animals. And this is going to have a lot of consequences for long-range signaling and synaptic transmission when neurons are communicating with each other and neuroplasticity as well will be affected. You've seen other experiments that show that in the heterozygous and homozygous animals, there's an increase in spontaneous spiking. Spike discharges are seen, and they're much more prevalent in the homozygous mutant animals. And then I mentioned synaptic plasticity, and this is an assay known as long-term potentiation, which is a commonly used method to assay long-term plasticity. It's sort of a substitute for learning and memory. And what is done is we measure the amplitude of a response in the hippocampus to exciting presynaptic neurons and then recording the postsynaptic response. And then we give a theta stimulus, which is a brief period of extra stimulation to the presynaptic neurons. And then there's a long-lasting increase in the response of the postsynaptic cells. And you can see that in the wild-type animals, there's a persistent increase in this activity that basically reflects a form of cellular learning and memory, and that's reduced, impaired somewhat in both the heterozygous and homozygous loss of function mutants. Okay, so basically what I've been kind of trying to emphasize is the point that it's kind of related to what Dr. Demarest said, that there's a mechanism of disease and there are ways to treat symptoms of a disease. And finding the underlying mechanism and treating that is really the best way to proceed. And as we just heard from Dr. Gray, you can have a lot of success by just simply replacing the lost SLC6A1, the lost GAP1 transporters, in his case with AAV gene replacement. You heard earlier this morning talk from Dr. Greenspan. His study with Dr. Demarest and others was just published that was looking at phenylbutyrate as a treatment for SLC6A1 disorder. And the impetus for this study, I think, really came out of work from a number of labs, including Caddy Kang's lab, that suggests that phenylbutyrate may help to be a chemical chaperone that increases the amount of GAP1 expression at the cell surface. And we were also looking at the effects of phenylbutyrate on neurons in the brain and also on SLC6A1 directly expressed in exogenous systems. And this is an experiment basically showing that phenylbutyrate application to a cell that's expressing human SLC6A1 causes, first of all, I should back up and say this is a voltage clamp recording. When we apply GABA to this cell, you can see there's an inward current that reflects GABA uptake, and there's a charge across the membrane that moves because of movement of two sodium ions and a chloride ion with GABA. And you can see that when we apply GABA, there's this inward current that reflects GABA uptake. But now when we add phenylbutyrate to the mix, you can see that there's a further increase in the current. And that occurs rapidly, and it washes out rapidly. And when we get, the current returns to baseline when we wash out GABA. And this change in current is also corresponds with a change in uptake of radio-labeled GABA in cells. Now I guess I'm being recorded by Siri or something. I don't know if there's a way to go back to. I can't move forward anymore. Can you check the PowerPoint? Okay. Okay, so what this means, what we interpret this as meaning that phenylbutyrate, in addition to its potential effects on trafficking and other effects that it may be having, also has this direct, what we would refer to as an allosteric activation of the transporter. And we went through looking at a number of analogs of phenylbutyrate to see what part of the molecule was responsible for this increase compared to control. And it turns out if you just get rid of the phenyl group and apply butyrate alone, you get the same effect, or actually even a bigger effect on GABA uptake. So both butyrate and 4-phenylbutyrate are positive allosteric activators. So this could be just coincidental, that they're doing the same thing, or it could be an important, at least one of the mechanisms by which phenylbutyrate works. Okay, so we went ahead and we've done some testing, and I'm just going to show you one or two experiments with butyrate. And what's shown up on the top left is another uptake assay where we compare wild type uptake of GABA, radioactive GABA into wild type heterozygous mice, and then into brain that's been superfused with butyrate at 0.3 or 1 millimolar. And you can see that if we add 1 millimolar butyrate to the uptake assay, that it restores the uptake in the heterozygous brain tissue to that of wild type levels. And in addition, if you remember, there's this impaired excitation spike coupling. And if we look at the effect of butyrate on the spike coupling, you can see that in the heterozygous mice, butyrate restores the spike coupling to wild type levels as well, which means that if you compare the control, these are the het animals, the control, and with butyrate, the efficiency of excitation spiking is restored to wild type levels. So we're excited to pursue the potential of butyrate as a therapeutic in this in SLC6A1, and we really also wanted to kind of understand better how it might be working as an allosteric activator. And one of the approaches that we've taken is you may have heard that this last year's Nobel Prize in Chemistry was awarded to researchers at Google, at DeepMind, who developed AI techniques to predict protein structures. And we utilized, and Google has generously made these protein structure algorithms open source. And so we've been using them to try to understand something about the structure of the GABA transporter in two conformations that we know must exist. What's shown here is the AI predicted model of GAP1, SLC6A1, compared to what we know from cryo-EM studies is the structure of the other main form of GAP1. So the transporter has to go through this conformational change where on the top is the extracellular domain, the bottom is the intracellular domain. There's a alternating access so GABA can bind to the outside, and then it undergoes this conformational change, and there's a gate here that closes, and that allows GABA to come out on the inside. And this structure has never been, hasn't been resolved yet experimentally. So the machine learning algorithm helped us to kind of get a clue about where butyrate might be binding in this outward-facing domain. And we used some other machine learning and molecular dynamics calculations in silico to predict where butyrate would be binding in that region of this outward-facing cavity. And both butyrate and, not shown here, phenylbutyrate are predicted by the algorithm to bind in the same place in the transporter. And so this provides a potential, now information that we can use as a hypothesis to start to ask what happens if we mutate residues in the protein around that binding site? Does that affect the binding of butyrate or phenylbutyrate? And also will help us to develop more different types of drugs that might work better than butyrate or phenylbutyrate. And you can see here that the machine learning algorithm and the molecular dynamics calculations predict that in the outward-facing conformation of the transporter, butyrate is binding in the same place as 4-phenylbutyrate. And you can see it's a distinct place from where GABA binds to the transporter, down here deeper. So it's interesting, butyrate and 4-phenylbutyrate seem to bind to this same pocket. And in addition, I'm showing here, there's a number of other analogs of butyrate that the machine learning algorithm predicts very accurately, at least qualitatively, to be able to predict the relative affinity of these allosteric activators. It's just another picture looking down on this pocket where phenylbutyrate binds. So how could that be affecting the transporter? Why would that make the transporter work faster? Basically, won't go into much detail, but we know that as I mentioned, there's this kind of conformational change. The outward-facing and inward-facing transporter goes back and forth, and GABA binds on the outside to the TO forms a complex, and then it undergoes a transition, and GABA is released on the inside. Now, we think that butyrate, because butyrate is preferentially binding to this TO form, is allowing the transporter to essentially work faster. Because we know from other work that this transition of the empty transporter is the slowest rate-limiting step in the transport cycle. And one way we can kind of test this idea is by using the inhibitor tiagabine you've heard about. And what tiagabine does is it binds to this TO state, and then it goes into a kind of a non-productive block state. Tiagabine doesn't get transported, it just stays bound in this pocket, and it goes into a different state. And what we can see is that what happens, so if you apply GABA to a cell expressing SLC6A1, again, you see this inward current because of the transport. Now when we add tiagabine, you can see the current now decays back towards zero. And what was really dramatic was that when we co-applied butyrate, that the entry of tiagabine to block the transporter was accelerated. And I'm not going to show you all this data, but it's on my student Kyle Cease, who's here. He has a poster on this work. So basically, conclusions of the work are that this S295L mutant we think is basically a good representation of a profound loss of function mutant, and of course, because every child that has a spontaneous mutation is likely to have a different mutation, and there's going to be a spectrum of phenotypes. There's going to be a spectrum of effects of these mutations, and so it's going to be important to understand what each mutation actually does in detail. We know that this complete loss of function mutation causes extracellular GABA to be increased by about twofold in the brains of het mice, which reflect the genotype of the patients. And as I mentioned, these changes that occur because of the tonic increase in GABA cause increased GABA signaling, affects neuronal spiking, affects neuronal communication and more complex synaptic signaling and synaptic plasticity. I showed you that the drug 4-phenylbutyrate, or Ravicti, acts as a GABA transporter allosteric activator, as does the over-the-counter fatty acid supplement butyrate. So we don't know whether this is the mechanism by which Ravicti's worked, and we heard today, earlier, there are very positive and exciting results from the Ravicti trial that just came out that suggest that it can be helpful, at least from the standpoint of ameliorating seizure disorders and EEG, restoring normal EEG patterns. So we want to look more carefully to see whether the fatty acid butyrate and related molecules might work the same way. And I showed you that the preliminary data suggests that it's worth looking at this in more depth, because if we treat brain tissue from heterozygous mice with butyrate. It restores normal GABA homeostasis. It can be returned to normal levels. So it's a potential therapeutic option for the SLC6A1 disorder. And another thing that kind of takeaway from the work is that we can use now AI and computational chemistry to identify this potential binding site for both phenylbutyrate and for butyrate directly on the GABA transporter, and further, refine the structure of the drugs that we can test to see whether they might work even better than butyrate does. And so it could create a useful drug development platform for SLC6A1 disease. And then finally, I want to acknowledge the team that did all this work. Kyle, who's here, he's got a poster. 2 other students, Andrew Voss, Ali Jester, and then another student, Andy Derlink. And then Greg Leary, postdoc, and collaborators at Lena Kalichev and Dave Hawley, and a collaborator at OHSU at the Volum Institute who are beginning to do more structural studies now of GABA transporter with Eric Guo. And I want to acknowledge funding from SLC6A1 Connect that made this work possible. And I think, as Steve said, I would concur that the reason I'm here is like 100% because of Amber. So I want to thank her especially. Thank you very much.